Thanks for the opportunity to talk with you all today a bit about my work. I'm really grateful and excited for that opportunity. So I am just starting up my lab. I've been there a little bit over a year. So I'm going to tell you a bit about sort of the foundation that I've built for my research program, along with some research directions that I'm really excited about that I'm building currently right now. So I suppose, like many of us, I have more questions than answers at this point. But I will try to tell you a few interesting stories and highlight the role of machine learning. As Scott mentioned, without further ado, here's the star of our show. This is Euplodes. It's a single-celled organism walking across a culture flask. This is more or less how I first encountered it. And when you see something like this through the microscope, once you figure out that it is indeed just one cell, I think it immediately calls to mind this question of how it is that something without a brain, without a nervous system of any kind, is able to coordinate this apparently sophisticated robot or perhaps insect-like kind of behavior. To really drive the point home, you already got a sneak peek of this. One of my favorite videos of all time. What you're watching is not a worm. It's just one cell that has attacked and is now in the process of devouring what's actually an animal. So these scenes are playing out in nature all around us. And these kinds of complex behaviors, of course, are important for the survival of microbes in the diverse environments they inhabit, but they're also important, for the function of our own bodies, like in development and immune system function. We've gotten a bit of taste of that in some of the other presentations. So the question really at the core of my work is to understand how cells control complex behaviors. And so I generally start out looking at a cellular behavior, looking at movement patterns, studying those intensively and quantitatively, then use perturbative experiments to try to get at mechanistic underpinnings of behavioral control. But I really want to understand not only what cells are doing in the lab, but what they're doing out in the world, how these behaviors play out in environmental contexts. And finally, I'd like to put all of this together in a comparative framework to understand something about how cell behavior evolves. So I think we're at a really exciting time in cell biology where we can take complementary perspectives to what we're learning in a lot of traditional model systems, use the unique biology of non-traditional model systems to address questions or gain insights that would be very difficult, if not impossible, to address using these traditional models. Okay. So if, like me, you're interested in principles of cell behavior, if you look across eukaryotic diversity, so this is a tree of eukaryotes displayed here, you're confronted with, I think, sort of an interesting, exciting challenge, but there's some hope and some opportunity. So the challenge, of course, is to find general principles tying all of this diversity together. Just to orient you a little bit, you're down here on this little branch, so there's a lot of diversity and complexity, but there's some hope, right? So the shared ancestry that's reflected in the convergence of all these branches is also reflected in a lot of shared cellular components and processes across all of this diversity. And we have also the opportunity, as I sort of alluded to already, of picking organisms that can uniquely help us address the questions that we're asking. And for me, this question of how cells control complex behaviors would be best addressed by a system that has a bunch of properties, complex behavioral repertoire that I can easily observe in the lab and poke at in the lab. A cell, for my purposes, is really amenable to rigorous behavioral analyses. Of course, it'd be nice if these behaviors emerge from core conserved components, and tools would be great as well. And finally, it would be great to have behavioral diversity across species. And indeed, as you may expect, Euplodes fits the bill. So you looked before at a cell crawling around in a flask. This is a cell in profile now. The cell's about 150 microns long, depending on the species. And you'll notice it has this highly polarized architecture. So there's a front and a back and a top and a bottom. Up the front, you can see this fringy little bit here. This is part of a structure called a membranellar band. And it's a compound ciliary structure. It beats those membranelles in a coordinated fashion to draw in bacteria and other small protists that it feeds on. Off the back of the cell, there are these little mechanosensitive bristles. And importantly for our purposes here, off the bottom of the cell, there are these leg-like appendages called cirri. And if you were to take a cross-section right through the base of one of those cirri, flip it over and look at it by electron microscopy, this is what you'd see. Each leg of the cell is composed of this highly ordered bundle of cilia. So these are your standard motile eukaryotic cilia and flagella. They have basically the same structure and function as any other eukaryotic cilium, like in a sperm tail cell. If you look really closely, so these are the basal bodies here, transitioning over to axonemes as you go a little bit farther away from the cell. If you look closely, you can see there are these linkages between the basal bodies. So within a cirrus, all those basal bodies are mechanically connected to one another. And if you look even closer off the edge of the cell, there are these little filaments that bundle together. And those all bundle together, and they're actually big enough to be seen just by light microscopy. So it turns out Euplodes was a favorite object of study of very early microscopists and early protostologists. And they saw all this complex cell structure. So this is just a drawing, a schematic from actually a PhD thesis from UC Berkeley from 1918, where they looked at this complex cell structure. So this is that membranellar band I talked about. This is actually the nucleus of the cell. And these fibers are depicted here. So these little circles are the bases of the legs. These fibers extend across the cell. They saw all this complex cell structure when they were looking at these cells back in the day. They thought animals must have evolved from something like a ciliate. They even went so far as to speculate that these fibers were part of a rudimentary nervous system. They call this the neuromotor apparatus of the cell. So we now know that animals did not, in fact, evolve from ciliates. This is one cell. It can't have a nervous system. But these early protostologists weren't imagining things. So this is part of the microtubule cytoskeleton of the cell. So this is from confocal imaging, the microtubule cytoskeleton of the cell imaged, it's labeled with sortubulin, this vital dye. So you can see there's a lot of complexity in this cell. And those fibers are indeed bundles of microtubules. And in addition to all that structural complexity, these cells have sort of obvious behavioral complexity as well. So this is just a little snapshot of some experiments that a bunch of different people have done, a bunch of students I worked with, where they put cells, Euplodes, in different prey conditions, in this case with a green alga, Chlamydomonas. So these different colors are just different movement patterns of cells over about 30 seconds in these arenas. And we can see as you change the prey density, so sparse prey, dense patches of prey, you activate these different movement patterns of the cell, basically different foraging patterns. You know, with patchy prey, you can perhaps most vividly see this yellow trajectory showing these local search patterns. With dense prey, the cells are mostly just standing still. They feed most efficiently that way. All right, so they have this complex behavioral repertoire in addition to sensing and responding to food. I really like this example where you can see mechanical sensitivity and also sensory discrimination. So I want to draw your attention to these 3 panels down here where you have cell-cell collisions. And you know from intro physics that these cells in the collision are experiencing equal forces, but you get very different responses from these different cells. So one hops away in surprise, and the other just keeps going as normal. So this is just a sort of simple example of this fundamental sensory discrimination task that a cell needs to make, which is to decide what it's doing to the environment versus what the environment is doing to it, so that it can avoid predation or keep going about its business if it just runs into a wall or something like that. Okay, so behavioral complexity. All of these cell movements are driven by movements of these leg-like appendages. So what you're looking at is a high-speed video recording of a cell running around on a cover slip. It's actually running around quite fast. This video is slowed down. This is all happening in less than about a second. The cell is reversing, reorienting, running off in a new direction. And it's doing all this while maintaining contact with the substrate without tripping over itself. So this, I think, suggests there's some interesting non-trivial appendage coordination going on here. And so while I'd love to understand all of that behavioral complexity, what I first decided to do is look at what I thought was the simplest thing that I could see cells doing, which is they will spontaneously just walk in a straight line. So this is just another view of a cell walking on a cover slip. It's slowed down a bit, so I hope you can appreciate these sort of stepping-like movements of these ciliae, these compound ciliary appendages, as the cell walks across a cover slip. So the question is, to what extent are these little legs coordinated? And if they are indeed coordinated, what does that coordination look like? And finally, where does that come from ultimately? Okay, so I mentioned these cells are amenable to rigorous quantitative analysis, and this is kind of what I mean. They really do have this animal-like body plan. They maintain this polarity. And so in this species of Euplodes, they always have 14 of these leg-like appendages in the same relative configuration. So what I can do is encode the stepping patterns as a series of these 14-dimensional binary vectors. So A through N, those are the 14 ciliae. Each entry gets a 0 if that cirrus is in contact with the cover slip, and a 1 if it's taking a step. And so a series of these encodes this sort of stepping patterns of the cell as they walk across the cover slip in more or less a straight line. Okay, so just to ground you in a little bit of intuition, and for what I mean by gait analysis. By gait, I just mean a stereotyped manner of walking. So if you're to watch a horse walk across a pasture, loping along, these rectangles here are depicting which hooves are in contact with the ground. You might coarse grain the stepping pattern into 8 different configurations of which hooves are in contact with the ground and which ones are not. So if you're to look at transitions among those coarse grained stepping patterns, these 8 gait states, if you will, you'd see these deterministic cycles as a horse walks around. This is characteristic of animal gaits. You have well-defined phase relationships among appendages. They're repeated cycles. This is kind of what defines a gait, more or less. So you might wonder, okay, it's a cell, it has no reason to walk like that, but many cells have these animal-like gaits with well-defined phase relationships. So these are some swimming algae studied by Kirsty Wan, where she's mapped the different gaits of these cells onto animal-like gaits. So you guys all saw some stepping patterns of euplodes. Do we think euplodes walks like an animal? You can probably guess. No, it's more complicated. So this is a representation of those stepping patterns. Time goes from left to right. Ceri A through N are the rows. Each white rectangle represents when the cell is taking a step. Okay, so you look at something like this. It's complicated. There aren't obvious phase relationships. But if you stare at enough of these, and even at this one, you might think, okay, but there's structure here. And indeed, there is some structure. So,
ask a question?
Yes, please.
These are longer, some are shorter. The longer ones, are they like ticking back and forth?
So none of these are. In this slow walking phase, it is, all of these are sort of a discrete activation and sort of a longer, generally a longer, slightly longer phase of quiescence, although not always. So these are actually single steps in this case. But for faster movements, that's not necessarily true. But in this case, it is. Yeah. But good eye, they are different phases, and that really is just sort of a different duration of recovery stroke, power stroke for the step.
What's happening physically between the foot and the substrate?
Hold on to that. I will show you. It's a really great question, and I'd love to know more about it, but I have at least one nice video to get us towards that question. If I forget to bring that back, ask me again. Okay, great. So, okay, this is going to be a bit information dense. And yes, please interrupt me at any time. I suspect this is relatively new for people. Love the questions. Okay, so we can look at all the configurations of serial activity and we can coarse grain. So for the aficionados out there, I used non-negative matrix factorization and dbScan to do your standard sort of PCA style dimensionality reduction and clustering. And I find that there are these 32 configurations of which ceri are active and inactive that the cells return to over and over again as they're walking along. Okay, so this is going to be a network representation of the gait of the cell. These nodes are those 32 configurations of which ceri are active and inactive. Size of the node is scaled by how often I see a cell in that configuration. Remember, if we were to play this game with a horse, we'd see something like this. And with euplodes, it's admittedly a bit of a hairball. Okay, so the edges of this graph now are transition rates, so scaled by how often I see that transition. This heat map colors are scaled by how much cell movement is associated with the gate state transition, with warmer colors corresponding to more cell movement. So just the feature to take away is there are a lot of movements that happen without very much cell movement. Okay, but there is structure in here, right? So the question is, to what extent is this sort of gate-like can identify cycles? And to begin to try to look for that kind of structure, appealed to an idea from non-equilibrium physics, broken detail balance. Okay, so detail balance, if you're not familiar, is a sort of complicated name for a relatively simple idea. That is a condition of equilibrium systems where transitions from one state to another will be balanced by the reverse transition, right? And so you can have fluctuations in your system, but you will see that balance across all state transitions. Systems that are not in equilibrium can display broken detail balance, which would just be that transitions are happening much more often in one direction than the other. And you can get these sort of cycles of probability flux in transitions in your system. And so if we look for significantly unbalanced transitions, they are indeed some of the most frequently sampled transitions for this cell. And so what's important to take away from this, right, we see these unbalanced transitions, the gate displays broken detail balance, is that there is a sense of directedness in time to these gate state transitions, making it somewhat gate-like. So to summarize the big takeaways from all of this, out of all those 2 to the 14 configurations of ceri being active and inactive in different patterns, there are only about 32 configurations that the cell samples. So 32 that are about, that are, each one is a bit noisy, right? It's not exactly the same 32 configurations, but more or less 32 configurations. We have, out of all the possible transitions among those 32 configurations, the cells are not sampling all of them. And in fact, there's even an identifiable directedness to sequences of gate state transitions. So it's complicated, but it is gate-like, and there are these sort of signs of coordination. So we can do a bit more work to try to look at what a sort of average gate cycle might look like. So I'm not going to go into all the details in the interest of time, but these nodes, again, are these gate states. They're numbered the same as they were in the other diagram. The arrows are scaled by conditional transition probabilities. So this is showing you the most likely transitions that you see from one gate state to another. And the colors of these nodes just correspond to, in blue, states that are both sources and recipients of these unbalanced transitions, and red ones that are just recipients. If you want to know more about this, there's a paper. I'll reference it in a couple slides. But the important takeaway here is that we can identify this kind of complicated cycle. Cells actually return to this resting state most often, more than any of the other states, in a given sequence of steps. And so the picture is that they'll relatively frequently return to resting state, no CRI active. And in the intervening steps, you will see these sort of unbalanced transitions. So there's this directedness to the sequences of steps that the cell is taking. Okay. The other important thing that I want you to take away from this is that I think that these kinds of representations are generally useful for phenotyping cells. They can help us gain mechanistic insights, and they can also be helpful for predicting cell behaviors. And I'm going to try to argue a bit for both of those points. Okay. So we saw all this gate complexity. And, a natural question is there any significance? Is there any functional significance to these complicated patterns? And to start to address that, we developed a really simple biophysical model of a cell that is this. So the goal of this simulation is to connect stepping patterns to overall 2D cell trajectories. So this is an imaginary third dimension, just to show you that we have two states for the CRI in our model. One, the cirrus is moving, generating a force in the direction the cell is facing. And the other state, this is 0, where that cirrus is in contact, acting like a simple spring, resisting movement. So we can sum up forces and torques and make predictions about cell trajectories from this simple mechanical model. And so if we calibrate our model and feed actual stepping patterns of cells, the simulation predicts they walk in straight lines. Great semi-quantitative agreement with our data. And so then we have the freedom to test the effect or make predictions about the effect of different stepping patterns. And so the two conditions we tested were we tape the same gate states and we just shuffle that sequential order, or we randomly activate and inactivate CRI, giving the same overall average serial activity level as normal walking cells. And the result is that with the shuffled gate state transitions, we predict cells have trouble maintaining precessive trajectories, but they walk at similar instantaneous velocities. And with this random activation and inactivation, cells walk really straight, but slowly. Okay, and so I don't have results from this yet, but one exciting research direction related to these simulations is I've been working with Agung Julius in the electrical engineering department at RPI to use reinforcement learning to predict optimal kinds of gates given different objective functions. So we can make predictions about gates that might be good for walking a straight line, turning, or gates that also might be good for cells that are confronting unpredictable fluctuations in the geometry of the environment, for example, or inability to finally control a step or not. And so I think that kind of approach, using machine learning, can hopefully generate testable predictions for experiments that might give us some insight into control principles. And I'm really excited about that as a sort of way to maybe get at fundamental control principles that are useful for controlling systems where we have high mechanical degrees of freedom, but limited fidelity in sensing and actuation. So like soft robots and that sort of thing. Okay. So
in these simulations, when you simulate the movement, is it like, does that depend on the gate configuration, or it's just a random direction where the entire cell like experiences a kick or something?
So it depends on the configuration, right? So instantaneously, so you have, the updating of the predicted motion of the cell is going to be a sum of the forces and torques that are a result of those spring-like interactions where the cirrus is not moving, and the actively moving cirrus, which is executing a power stroke in this model, generating a force in the direction the cell is facing. And so this is an imaginary third dimension. In the actual simulations, we have this 2D configuration of the CRI in the relative configurations that we find them in the cell.
So if I understood correctly, each CRI node has a self-propulsion vector or something like that.
Yep, that's right. Great. Okay. So
Ben, I'm sorry if I missed it, but do you need all of the CRI?
What do you mean by need all of them?
Is it required for moving in a straight line?
Okay, so I don't know because I haven't done the experiment and I haven't tested simulation robustness to changing numbers. One thing that is the case is that in euplodes, across all species, there are about 100 described, and they all have either 14 or 15 cirri within the genus. I suspect that comes more from a developmental constraint than a motility functional constraint. I would love to do experiments where I'm ablating individual cirri. So I don't know how important they are. Just given intuition and looking at the dynamics, so some of the cirri move a lot more than others, and that becomes even more biased when you start to deviate from this slow, straight walking. I suspect there are some that are going to be more important for certain kinds of movements or for maintaining these straight trajectories. So just one, I guess, quick little instance is that these two cirri, F and G, have very highly correlated movements. And if you watch enough videos, you do get the sense that the cells are reaching out and adjusting their orientation using those cirri. But that's pretty speculative. Yeah. Yeah. Okay. Great. So we wanted to understand, how this coordination might be accomplished. And one of the obvious things to start poking at is this structure in the cell that's associated with the cirri. So this is a 3D reconstruction of that neuromotor apparatus, these bundled microtubules associated with the cirri. So A through N, those are marking the base of all those 14 cirri. These different colors are just morphologically distinct classes of fibers. In cyan, just thicker fibers that tend to be straighter, and in magenta, thinner fibers with a bit more curvature. Okay, so there's this complex structure. These fibers do apparently interconnect with one another. And the question is, if we perturb this fiber system, can we perturb gate coordination in an interesting way? Okay. So one thing that I was pleasantly surprised to find is that when I treat the cells with microtubule inhibitors, I see very distinct motility defects. So these are DMSO control with nicotazole. This has the effect of depolymerizing microtubules. Notice these cells have a lot of trouble maintaining precessive trajectories. We can also hit them with paclitaxel or Taxol and see a different motility change. These drug treatments have all the effects that you'd want to see, dose dependent, they're reversible if you wash them out. Okay, so the affect motility. And we can play the same game now and look at patterns of gate coordination in these perturbed cells. So one important thing to note is that we see these defects. Also, if we look at the fiber system, in, for example, nicotazole-treated cells, there's a clear effect on the fiber system. So we lose a lot of the magenta fibers, and the overall fiber system itself is shorter relative to the length of the cell. So there's this geometrical effect and possibly topological effect on the fiber system. And if we look at patterns of gate coordination using the same approach that we did in the unperturbed cells, we see the dominant effect is in sequences of gate states. They still seem to sample the same configurations of which cirri are active and inactive, but what's really broken is that sequential logic of the gate. So now that, remember, hit a targeted prediction from our simulations, which is that if you mess up the sequential order, you have trouble maintaining precessive trajectories. And indeed, these cells actually do walk at comparable instantaneous trajectories to the unperturbed cells. So that was a nice bit of consistency. Yeah.
The cortisol treated,
yeah,
there's still a lot of tubulin.
Yes.
Because those are axonal and stable.
So I don't know why so much tubulin remains. My assumption would also be that it's more stable. I was actually honestly very surprised that the fibers were affected because there are these huge bundles of microtubules. I expect those to be highly cross-linked, stable populations, but based on the drug treatments, they must be dynamic. I would love to just know more about the structure of those bundles. So the ones that remain, it could be that they're more stable. It could also just be that nicotazole is not as effective on these cells. We know that their tubulins are divergent to some extent. And so this is one difficulty in drug treatments with these protists or these non-traditional systems. We don't quite know if it will have the most comparable effects, but this is just to say that it does have an effect. It has all the features that you would expect it to have, but there's a little bit of subtlety in interpretation. Good question. Okay, so one nice thing about these cells is that they're big enough that we can do experiments that would be very hard in other cells. One of those is microsurgery. So you can actually cut these cells. And this experiment is actually reproducing experiments that were done in the early 1900s and then repeated in the 1960s where people got supposedly opposite results. In the early 1900s, they argued that you cut cells, you cut those fibers, you mess up gait coordination. In the 1960s, they said, nope, everything's the same. But no one had actually looked at walking cells with cut fibers. And what I was really happy to find is I can cut cells even all the way in half. And this is the back half of the cell. In about 12 hours, they regain motility and they actually will walk. And so this fragment's going to find its footing in just a moment and walk around. And there it goes. Having trouble, but it finds its feet, it walks. Okay, and so that trajectory below is the trajectory of the piece. It, obviously has trouble walking. But I can do an experiment where I compare cells that I've just stabbed with needles, wounded grievously, to these pieces. And what I see is gait defects that are actually very similar to what we see in nicotazole treatment. So this is slightly counterintuitive. But if you mess with gait coordination, you mess with the fibers, you hit these cells with microtubule inhibitors, one of the dominant signals of this defect in the gait is that you see more synchronous serial activity. And so that seems to be somehow problematic for these cells walking in straight lines. But I think that does suggest an interesting sort of principle of gait coordination. So at the end of the day, we think that these fibers are working somehow, and I'd love to know how, to mechanically mediate gait coordination. It has to be happening on very rapid timescales, so mechanics is sort of an obvious candidate. And it seems to be in some sort of inhibitory fashion. So the mechanism really seems to be to stop the cell from taking the wrong sort of step at the wrong time. And that's at least a little bit reminiscent of neural control of gaits in animals, where you have these mutual inhibitory neural connections that gate or block a limb from moving while another one is moving during limbed locomotion. But another sort of interesting feature of this microtubule scaffolding dictating cellular coordination is that it's a principle that you see quite broadly across eukaryotic diversity, or at least hints of it across eukaryotic diversity. So in Chlamydomonas, an organism that has been studied quite intensively, it's known that there are these mechanical connections between the basal bodies that are important for ciliary synchrony in swimming. You also have in another ciliate tetrahymena, and also in multiciliated cells in animals, these microtubule scaffolds, and it's known that those affect patterns of ciliary coordination as well. The hypothesized mechanism there is mostly that it allows more freedom in misalignment of these cilia, but there may be some more interesting mechanical coupling and mechanisms involving mechanical coupling that we might have a unique window onto with euplodes, right? We have these distinct patches of cilia that have sort of unique dynamical identity that's functionally important for the cell. And so I'm really hoping to dig more into mechanistic details there. Scott, how much time do I have now that we're off the schedule?
I mean, you have a few minutes. I mean, you probably like 10 more minutes.
Okay, great. Okay, great. I have a few more interesting directions, but I'll keep it in that timeline. Okay, so here's your intuition. We have geometry and mechanics that dictates dynamics in our cell, maybe a bit like these fantastic kinetic sculptures from Theo Jansen. These things are just made mostly out of wood and a little bit of fabric. And by virtue of the mechanical linkages, they are able to coordinate their gaits as they walk across the sand, in this case, driven by the wind. Okay, so this is perhaps more broadly a principle of behavioral control across eukaryotes. So with euplodes, I think we have this really fantastic system to integrate across scales of biological organization, connect these molecular mechanisms with behaviors that cells are making in different sorts of environmental conditions. Okay, so now I'm going to tell you a bit more about directions that I'm currently excited about building in the lab. So one of those, as I alluded to earlier, is looking more broadly at the overall behavioral repertoire of the cells. And here's where machine learning becomes very important. So to build up the kind of data set that I need to really do a full analysis to try to figure out what are all the different kinds of behavioral degrees of freedom accessible to these cells, really need a lot of automated tracking and automated identification of these sub-movement patterns. And I've got a fantastic student, computational biology student, Alex, who has started to work on certain aspects of this, including identifying different sub-movement types, and then working with automated tracking to look at all the different details of appendage movements associated with these different cell movements. So if you do machine vision stuff, you'll know this is a really hard tracking problem. And what Alex has done is taken software, deep learning-based segmentation, or rather deep learning-based tracking software that people have developed for animal pose estimation. This particular one is called SLEEP, And he's been able to adapt those slightly to our purposes for these microscopy videos. And he can now automatically detect serial movement patterns in these videos, even when you have quite complex serial movement patterns. So ultimately, we want to connect all the details of those appendage movements to submovements of the cell and understand how those submovements are organized into functionally relevant behaviors, things like finding food, avoiding predators, and all that fun stuff. So in addition to looking at just these overall behavioral patterns and looking at what the behavioral degrees of freedom really are, we want to get at mechanisms too. And so this is just a fun additional mechanism to introduce you to. So it's not just the microtubules that are controlling things. There are these signaling processes in cells. So this is fantastic work from Alice Hernissen that Caitlin's now working on extending, where she treated cells with caffeine. So this cell that looks like it's breakdancing isn't actually breakdancing. She's registered the video. So the cell is actually hopping around dramatically on a cover slip. And that's what the normal sort of avoidance reaction of the cell looks like. It's doing something called a sidestepping reaction, where the cell hops backwards in a shrimp-like fashion, reorients, and runs off in a new direction. That's also what they were doing when they experienced mechanical stimulus. So it's part of this avoidance reaction. When she treated cells with caffeine, they had a lot of trouble accessing that sidestepping reaction. They're almost stuck in a certain subset of these gate states, perhaps. And so the mechanism, most likely, and she's tested this a little bit, is that caffeine is a broad phosphodiesterase inhibitor. It's probably leading to increase in cyclic AMP and cyclic GMP. These are known regulators of ciliary activity. And indeed, when Alice treated these cells with cell-permeable cyclic GMP, it phenocopies this caffeine phenotype, suggesting that elevated cyclic GMP is what's behind this. Downstream, what's probably happening is that cyclic GMP is acting on cyclic nucleotide-gated ion channels that eventually are modulating calcium levels in the ciliary. So calcium is sort of one of these major regulators of ciliary activity broadly across eukaryotes. Okay, but the picture that I have of gate control is that there are these two modules that act sort of in concert. And one involves signaling. I mentioned there's ionic signaling in the cell, so there's fantastic electrophysiology where people have linked electrical activity to details of serial or movement patterns in Euplodes and some of its relatives. And so I think that acts in concert with this cytoskeletal activity that's modulating details of stepping patterns. So here's the picture. You have this signaling that's setting a disposition. Steris is going to try to beat at a particular kind of frequency, perhaps with a particular kind of waveform. And then the geometry and mechanics of the cytoskeleton is going to constrain that attempted beat frequency, stopping the cell from taking the wrong sort of step at the wrong sort of time. And I think that these sorts of coarse-grained state transition pictures of gate coordination are going to be helpful for us to disentangle some of this complexity. So the picture kind of is that signaling is setting the spectrum of possibly accessible gate states. The cytoskeleton is tuning the transitions among them. And that's at least consistent with the data we have so far. Okay, so we're going to do a brief interlude.
One quick question.
Yeah.
So do you assume that these transitions of different ways of walking, they don't depend on the history?
They do. So this is the fun feature of them. So yeah, that sort of Markov style picture is a little bit misleading in that there is some history dependence, frustratingly non-Markov. And so, there are ways to deal with that, right? Coarse grain in different sorts of ways, but
Large language model?
yes, this would be another good use for machine learning to try to figure this out. And so one approach that we have taken to try to address the apparent non-Markovian nature of these gate state transitions is to use an algorithm called causal state splitting reconstruction that basically generates a hidden Markov model, and it's supposedly the minimal, maximally predictive model for your system. An issue that we have with that is like many of these really beautiful, sophisticated mathematical constructions that people have, it does not deal well with noisy data. And so it's been hard to get results that we can really work with, but maybe with an expanded data set, that will be a bit more tractable, but it's a good idea. Yeah, question.
These cellular organisms,
Yeah.
do they experience fluid anytime?
Yeah.
And are you challenging them on the substrates as well?
Yes. So we are definitely looking into the effect of substrate roughness, stiffness, composition. They're always embedded in fluid. So these things are aquatic organisms. There's always fluid around them. I've played a little bit around with viscosity. They seem surprisingly sensitive to viscosity. So if you increase the viscosity, they have a lot of trouble settling down onto a substrate. Yeah, good question.
Can I ask?
Yeah.
one more?
Yeah.
You play with external stimuli, like give them something and see.
Yep.
Look at the same.
Exactly. So we're just building up that understanding now, and they very quickly and clearly respond to things like prey cues in the environment. There actually is a kind of sophisticated mating behavioral repertoire, and you get these big swarms of cells that can form. And so there's a lot of sort of directed motility. And one nice thing about protists is that you often are relatively easily able to interpret, functional significance of these kinds of behaviors. Like the cell is running towards food, it's avoiding danger. And so we're building up that kind of understanding of the overall behavioral repertoire of the cell. Yeah. Great. Okay. So a brief interlude, and then we're going to do one more quick story about euploides after that. So I mentioned that I think that these sort of perspectives on cell behavior are useful. So I decided to put that to the test with another fantastic organism. So I've started to play around a bit with predatory amoeba. So what you're looking at, this paramecium cell right here, paramecium multimicronucleatum, it's about 250 microns long. So this is millimeter scale cell. This is just its pseudopod, phagocytic pseudopod, where it's capturing this prey item. This incredible predatory behavior of these giant amoebae. And if you look at enough of these cells, watch them running around, you distinctly get the idea that you can kind of interpret their behavior, and you can even predict what they're going to do. And this is pretty consistent with some historical observations. I would highly recommend, if you're curious about cell behavior, to look at Herbert Spencer Jennings' Behavior of the Lower Organisms. Great collection of interesting observations of cells. One sort of cell that he looked at was amoeba, and he saw that they had these sequential hunting patterns that they would use. And so I've taken this morphodynamic approach. These things are very easy to segment. No fancy machine learning needed here, just intensity thresholding. You can get a bunch of shapes of these amoeba and ask. So this amoeba at the center here is hunting, and you're going to see another one in this sort of migrating mode. So they have these distinct behavioral states that they seem to embody, where they can be feeding. If you put a prey item next to them, they're going to pounce on it and eat it. But when they're in this sort of migratory mode, that one that zooms by, they're insensitive to prey. And the question is, can I look at a relatively short sequence of cell movement or shape patterns and predict what it's going to do given an environmental stimulus? And so we've already seen people taking different morphodynamic approaches to cell prediction. Here, okay, we can do PCA after we've extracted a bunch of different shape parameters of these cells, classify these different amoeboid postures, if you will, or different shapes of amoeba. And if we look at the postures and the transitions among different postures of these cells, what's interesting is that when you have migrating and hunting cells, they adopt similar postures, but it's really the characteristic transitions among these different postures that allow you to very easily distinguish between these cells that are hunting. If you put a prey item, it's going to pounce on it. Migrating, it's going to be insensitive to prey. And so that got me wondering about, if you can use these basic kind of approaches for other cellular predictions where the behaviors are a bit more inscrutable. And that led, Christina Braverman is working on the amoeba problem, but she's also teamed up with Jen Hurley's lab in the biology department to look at macrophages. And the question is, we know these macrophages can exist in these different inflammatory states where you have different molecular and functional signatures. Can I look at these cells running around and decode that inflammation state from movement patterns? So this video, I really love that macrophage seemed to have eaten some, schmutz on the cover slip. It looks just like an amoeba, but actually it's a bone marrow derived macrophage. And so this is, I wanted to bring this up because I think there's one more interesting use of machine learning that people haven't really talked about so much, and that's students using large language models to help them code. So Christina is a brilliant experimentalist. She has industry experience, really great on that side, very little computational experience when she got into my lab. And she's been able to put together this computational pipeline, mostly using cell posts for segmentation and then extracting all these shape parameters. And she's been able to write all of this in relatively short order. She's just been in the lab a number of months, largely through assistance from these large language models that help students that have very little coding background really jump in quickly to do rather sophisticated coding. And she's already been getting some really exciting results. that are at least consistent with molecular and functional data on macrophages, where she does seem to see these morphological, perhaps morphodynamic signatures of inflammation states. But stay tuned. We're just starting that. Okay. So do I have 5 minutes or should I be done?
It's 2 minutes.
2 minutes. All right, 2 minutes. So the final direction, I mentioned I'm interested in these comparative approaches. So I can make use of diversity of Euplodes species in the different environments they inhabit. That diversity and my interest in understanding cell behavior in the wild sometimes brings me out into the field to make observations, see how they traverse different environments. So I'm going to skip through a lot of the details, but this is just to say that there's some cool morphological and behavioral diversity that you see in these cells. So this is a previously undescribed species of Euplodes called Euplodes gigatrox. This is a clonal population. They're all the same kind of cell. You'll notice a very different behavior from this big one in the middle here. They become these supergiant cannibals. And so here you have this linked morphological behavioral transition. And so I'm trying to understand this relationship between cell structure and behavior within these species. And I'll skip just to say they're fascinating. There's a lot of morphological and behavioral diversity. This is a preprint, so you can read the story if you want. And just to say that we have a bit of a zoo in the lab. And so you do see behavioral and morphological diversity across these species. We can take a look at these different fiber systems. And I would love to have enough of an understanding of how this fiber system might be acting to coordinate appendage movement, to predict patterns of appendage coordination, perhaps even cell movement patterns just from structural information. So we know that it's very, very hard to take a eukaryotic genome and understand something about the behavior of that cell, especially if it's a weird one like Euplodes that has tons of unannotated genes in the genome, even if you have a high quality genome from this thing. But we're getting much, much better at getting structural information, looking at cytoskeletal information. And in a lot of ways, cellular diversity really does stem from cytoskeletal diversity. So this might be a nice window onto cell behavior, as well as sort of a test of my understanding and ability to predict cell behaviors. And across ciliates, we really, I think, have a beautiful, vivid illustration of a theme that's more broadly true of eukaryotes. And that is that across all these cells, Euplodes is here, these cells are all drawn to scale, there's incredible diversity of form and function. But synapomorphy of all of these cells, so a shared characteristic that was present in the last common ancestor, is this ciliary scaffold of microtubule constituents. And it's really by virtue of arranging and rearranging that scaffolding that you get all of this cellular diversity and associated behavioral diversity as well. I think that's broadly a theme across eukaryotic diversity. And I'd really love to understand the extent to which there might be principles dictating, perhaps even constraining all of that behavioral and morphological diversity and that integral relationship. So with that, I will thank fantastic students and postdoc who have joined my lab, great collaborators, RPI for being a wonderful place to be starting my research program, and to thank you all for your time and attention. All right, cell feet. Let me get us there. Distinctly creepy feet of the cell. Okay, so this is from, this is the actual foot of the cell. This is the foot of the cell. This is from a technique called interference reflection contrast microscopy. We're going to see in dark things that are very close to the cover slip. What happens is this cell is going to get struck by another cell that's out of the field of view. It's going to engage traction. And what I want you to appreciate is the contact area of the foot. It gets really big. So I think they probably are using the gecko trick, right, where you have these low affinity, non-specific high avidity interactions, increases that contact area, sticks down, and then also gives it this non-specific adhesive capability and rapidly reversible adhesion, where to lift its foot off, all it has to do is close and peel to unstick. But I would love to functionalize some surfaces and test, to what extent is this van der Waals and this capillary. I don't really know anything about the nature of the adhesion, but I'd like to know more.
Very briefly, maybe I missed it. Are there like dyneins and kinesins? How do they swing those?
Yeah, so it's eukaryotic cilia, so it's dyneins that are driving the bending of these. Yeah, and so some of the details of that regulation are certainly going to be different, but calcium is a major regulator of that dynein activity, certainly in these cells. Yeah. I guess I saw your hand first, sorry.
So I mean, can you do a dimensionality reduction by chopping off some CI?
Yes. So yeah, this is what Radhika was asking about as well. Would love to do those ablation experiments. I haven't found a clean way to do it. It's so in a lot of cells, you make them unhappy in really any way, and they'll just drop their cilia. So Chlamydomonas does this.
So it's not lethal if you chop some of the cilia.
A lot of cells will just drop them in stressful conditions. I can't figure out how to get these cells to drop them. I've tried a bunch of different drug treatments, stress treatments. They're really robustly scaffolded in. So maybe laser ablation is the way to go. They are able to heal pretty well. I also might be able to figure out microsurgery. It's just kind of hard to manipulate the cell. And so what you have to do is kind of like hold the serous out with a suction pipette and then cut with the other microneedle. And I haven't been able to figure out how to do that yet. But Jing, I saw you next.
Two quick questions.
Yeah.
One, do they have like a rigid cell wall? They seem to hold the shape.
Yeah, so they hold their shape. It's not a cell wall, it's microtubules. So there are these sheets of microtubules at the cortex of the cell. And that's a lot of what gives it that kind of structural integrity. But they maintain that polarity. They're at the scale of a cell, they're relatively stiff.
The other question is like, how does this very fancy machinery, how does it kind of scale with size? I mean, what's the range of size that you have observed?
So across Euplodes diversity, the smallest species are about 25 microns long, and the biggest ones are 150 to 200 microns long. So that's the cell scale. Across that size range, you also have either 14 or 15 ceri. But interestingly, across several genera, maybe dozens of genera actually, you have a lot of walking cells that range from maybe 10 micron size scale on up to, about, I guess, maybe 500 microns are the biggest ones. The length of the ceri are like 10 to 20 microns long usually. Yeah. But it's interesting. So I haven't done detailed scaling analysis, but a lot of the freshwater species seem to have longer ceri relative to the cell body length. And the marine ones have shorter, dense, like thicker ceri proportionally. And maybe it's related to viscosity. I don't know. That would be wildly speculative. But there's interesting scaling that seems sort of nonlinear.
Longer, and so the overall like rigidity would be very different.
Yep. Yes. And I do wonder if the mechanics are tuned to their environments. Absolutely. Okay. So many people.
Take like one more question.
Okay.
And then we can discuss.
I think I saw. Well, you've asked questions.
Questions, you're right. Yeah.
All right. Go for it. So I hand it back. Yeah. We can talk later.
So,
Yeah.
Would you be able to, like, I don't know if I missed it or not, but would you be able to image it on a side view microscope, sort of to get the much finer motion of these?
Yeah. You would in principle. I had thought about setting up that kind of microscopy setting. It doesn't require too much finagling. I just haven't done it, but it would be interesting to have actually a dual view.
A dual view would be much better.
Because you could do full 3D. And there are people actually, there's this German group that in the 90s was doing that kind of imaging of a relative of Euplodes and got really fantastic 3D movements.
Perhaps
of ceri.
follow up on that, I think, on this topic is that, did you do any mechanical squishing of this just to see what it does? And sort of like much more lethal sort of?
So I haven't done too much confinement beyond putting them in sort of naturalistic conditions where they kind of get stuck in pockets and stuff like this. So I don't know what would happen, but
or confining them by microfluidic channels just to see.
Yeah, would be
how
interesting.
persistent they would be in one.
Would be interesting to know, but I haven't done it. Yeah.
I mean, along the same lines, it would be interesting to set up like a maze-like architecture and make them navigate through that.
Try to get students to do that. Yes, I really want to. We do have some pretty good microfluidics folks at RPI, so I'm hoping I can find the right collaborator. I would love to put them in mazes.